So the background to this project goes back a number of years now, and in pediatric acute lymphoblastic leukemia we have been using risk factors for probably about 20 years now in order to stratify patients into different risk groups and then receive risk adapted therapy. And the number of risk factors that we've been using have been between 5 and 7 depending on which clinical trial we're using. But what we've always done in the past is use risk factors as binary variables. So patients either have or do not have a particular risk factor. Now, for some risk factors, this makes logical biological sense. However, for many risk factors, like minimal residual disease and age and white cell count, using binary risk factors doesn't make biological sense, because you have to create a threshold at which one patient is either under 10 or over 10 if you use age as an example. So what we've done in this study is move away from the use of binary variables to use continuous data. And we've done this on a trial called UCAL 2003, which is a trial that ran between 2003 and 2011 and recruited over 2,500 patients. So what we did is we pooled all the risk factor data that we had for that, using wherever possible continuous variables, which gives us much greater granularity in order to be able to predict outcome. And we developed a prognostic index. And this prognostic index in that initial discovery cohort was very powerful at predicting relapse. Obviously, what we needed to do is validate that in independent cohorts. So what we then did is we teamed up with 3 other clinical study groups from Scandinavia, Holland and also Germany, and validated that prognostic index in a similar size validation cohort of about 2,500 patients, and it validated extremely well. There was some variation which would be expected, because you're unlikely to identify a prognostic index that's absolutely perfect. But the variation seemed to be in patients that were interestingly at the highest risk of relapse. But that's kind of logical in a way, because if you look at the various treatment protocols, that's where their treatment varies most significantly. So what we've been able to do is develop and validate this prognostic index. And the real advantage of having this numeric prognostic index is it gives you a completely different way to design future clinical trials. You can now identify and define a good risk cohort of any size you want, depending if you want the best performing 50% of patients or if you only want patients that have a relapse risk of less than 5%. So it gives you much greater flexibility in order to be able to design clinical trials or study protocols in the future to identify groups of patients of the size you need for your treatment intervention, and also to have a homogeneous outcome. And it's much greater granularity. And what we further did is we tested this prognostic index against the risk groupings that were happening in each of the individual 4 trials, and proved that this prognostic index increases your power to predict relapse by an order of 10 to 15% in every single clinical trial that we tested it on. Yes. So it's a relatively simple index. It only requires you to know about the minimal residual disease at the end of induction, a few key genetic abnormalities, and your white cell count. So it's relatively simple. The actual equation to calculate it is quite complicated, but the idea is you would use a kind of online tool, and you would just type in the various variables, hit a go button, and it would tell you what the prognostic index is. So it would be, it's for a computer, it's a very easy formula. For a clinician sitting in a clinic, no, that wouldn't be the way to go forward. But what we'll do is we'll put it online, and then people can calculate this prognostic index.